I'd like to thank Todd Constable for coming up from Yale to give the talk today. Todd is Canadian, and you grew up in Winnipeg, right?
Yeah.
Grew up in Winnipeg.
Winnipeg.
And so, I grew up in Minneapolis, and Winnipeg is like Minneapolis, but worse. I feel a certain kinship because of that kind of Midwestern North American, the cold part of Midwestern North America. Todd is not a psychologist. He is, his degree is in medical biophysics. So he started off in physics, and then he got his PhD from the University of Toronto in medical biophysics. And he is now the director of MRI Research and co-director of the Magnetic Resonance Research Center at Yale. So he brings a really fresh perspective onto functional neuroimaging because of his background. And so I think that what he's going to present today is kind of radical, actually. At least, I happen to agree with it, but I think that you've hit some resistance with this perspective. And I have great respect because he's gone back and really looked at the assumptions that underlie a lot of what we think is ground truth. So what some of us might think is ground truth in functional brain organization, and gone back to the original literature to see what the real foundations are and why we should be questioning that. And I'm going to let him tell us the full story.
Great, thanks a lot. It's great to be up here. Thanks for having me. I've always heard a lot about Dartmouth, but I've driven by, I think, but I've never actually stopped here. So it's great to be here. Yeah, hopefully what I'm going to say is not only radical, but right. I don't actually know that, though, but I think it seems to make sense. And actually, I've already given this talk to most of the people in the room here in our one-on-one conversations today, so I'm not sure how much new you'll find here. But so what I'm going to talk about today is fMRI and some of the assumptions of fMRI. And we'll talk about resting state bold signals and then maybe the not so resting state, so when we apply a task to functional MR. And hopefully I'll give you a little bit of insight into what I think we can learn about functional organization from functional connectivity. And then I'm going to get into node definitions and atlases and the Connectome. And that's going to lead us into kind of like what are the goals of human brain mapping. And we, a lot of you already heard some of this in our discussions today. And by the way, thanks for all the discussions today. It was really great. I really enjoyed all my conversations. And it's cool to get feedback and insights and to see what your responses are to some of these things. So I assume everybody here knows what functional connectivity is, but I have like one slide here to talk about that. We're really looking at resting state bold signals that are correlated in time, and this indicates that things are functionally connected. Actually, even though this is, and so the first paper on this was Biswal in 1995. Biswal published this paper in 95, and basically from 1995 to 2005, people were all focused on physiologic artifacts and saying this isn't real. This is like, there's coherent activity because the cardiac pulsation and things like that, and that's why it's symmetric in the brain. But what Biswal did was they did a finger tapping experiment, and that's what's shown in A there. And then they took a seed region and one of the motor cortices on the right, let's say, and they said, what's this connected to in the whole brain, just by correlating that seed region with the rest of the brain. And lo and behold, they pulled out the motor network. And so that kind of started us on this whole road of functional connectivity. The reason I put this up, and I want to emphasize that I'm talking about functional connectivity, is that I'm going to talk about the flexibility of the brain and kind of the flexible organization. And in submitting several papers on this, I've hit a lot of resistance from people that feel like the brain is structurally defined and that what are you talking about when things are flexibly connected and stuff like that. And so I want to emphasize it's functional connection. Neurons aren't jumping around from location to location, which is what one reviewer suggested we were suggesting. And this is just things are synchronized or not. If you're that reviewer, I'll forget it. So this is different. This is distinct from task-based fMRI. So in task-based, you might have an external stimulus, and you look for a response that looks like that blue line, and that is your activation. And so you're looking at typically a few regions that are synchronized with that sort of onset of the stimulus. In connectivity, there doesn't have to be a stimulus. There can be a stimulus. But in connectivity, we're just measuring bold signal, this blood oxygenation level dependent contrast. We're measuring that over time. And regions that tend to oscillate together, this is low frequency, like 0.1 hertz or less. Regions that oscillate together tend to be functionally connected. And so that red and the green area, we might say are functionally connected if they're highly correlated. The cool thing about this is that you can extend this to measure the correlations. If you have an atlas like what's shown here, you can look at all combinations of connections between all combinations of pairs of nodes and come up with a connectome, the functional connectome. And so in the original Biswell paper, it was kind of a seat region to the whole brain. But now what I'm mostly going to focus on is looking at the connections between regions or nodes and building up the connectome. So this is the functional connectome. And there was a whole project called the Human Connectome Project, which was out of WashU, and they imaged 1,200 people, and they were trying to get these connectivity matrices, which are formed by numbering the nodes, let's say 1 to 268. This is one of our 268 node atlases, and you look at all combinations of node number 1 with all 267 other nodes, and you can fill in then the correlation value, and that represents how well connected those nodes are. There's a couple of interesting features here. One is that this matrix is symmetric, and that's because we don't have any directional information. So 1 to 5 is the same as 5 to 1. If we had directional information, that matrix wouldn't necessarily be symmetric. But for now, we just assume everything's symmetric because we don't have directional information. And there's really two ingredients we need here to do this. We need an atlas, and we need the time courses, and then we can get the connectome. And so this is your functional connectome. I think the functional connectome is fascinating. This is actually what it looks like for a 268 node atlas. So that's a 268 by 268 element matrix. So in that, there's 35,000 connections. And I was asked today what I think is cool. And what I think is cool is that there's a ton of information in there. And what I'm focused on these days is how to extract that information. So yeah, by the way, it's 35,000 nodes or connections, edges, for that 268 node atlas. For something like the Glasser atlas that's 360 nodes, there's 70,000 unique edges there, connections between nodes. And so there's potentially a lot of information there. We're only looking at the bottom half, but when I say 70,000, that's in the bottom half. And there's been a lot of work already. So with Emily Finn, who's coming here as faculty next year, we did the functional connectome. So that's her little fingerprint picture up there, where we showed that this was unique to an individual. So we could identify you by this matrix. And that was, we didn't set out to look for that. We were actually looking at grouping. We were looking at trying to cluster people by brain state. And in fact, in the HCP data, they had all these different tasks, and we were going to see if the working memory task all clustered together. But instead, an individual would cluster together across all tasks. And so it turned out that you could actually identify individuals with this. Yes?
So is this already, have you gotten rid of the hemodynamic response? Is this?
This is just that correlation over time, and you replace.
You can get just correlation from the blood, right?
Well, it is a blood signal. You're looking at changes in blood. The bold signal is the change in local tissue oxygenation, which causes a T2 star change, which is a complex function of flow, volume, oxygen consumption, and oxygen demand. So that leads to this bold signal. We look at two regions and how their bold signals are correlated, and we replace that with one number, which is a cell in this matrix. So whether we get rid of blood flow, not,
I mean, if you get rid of that,
it's an intrinsic signal.
Yeah.
There's various pre-processing you can do in terms of global signal regression and stuff like that, and I'm going to ignore all the pre-processing steps. But there's that's what the first 10 years of connectivity was from 1995 to 2005. But I think these are real signals, and we can get, real results from that are encouraging. So Emily showed that this was unique to the individual, and then as part of that paper, she showed that you could build a model from this data to predict fluid intelligence. And Shilin Shen in 2017 published additions to that model, and we have a Nature Protocols paper there. But then there's been other work on predicting fluid intelligence from the Connectome. There's been work by Monica Rosenberg and Marvin Chan at Yale in terms of looking at attention networks. Autism symptoms is a paper I had using the ABIDE data and predicting social responsiveness scores or ADOS scores. And then there's some personality work also from the Yale psychology group. And then reading recall, which Emily's on this paper, or no, Monica's on this paper, on could you predict reading recall from the matrix that you obtain when somebody's reading a passage. So I think there's a lot of information in here, and one of the things I'm interested in is building models to get that information. And so how do we do that? So in summary of that part, the functional organization seems to be unique to the individual, and it seems to be relevant in the sense that so we can identify people, and these functional phenotypes seem to tell us something about the person. And one of the things I'm particularly interested in is, can we categorize patients with this? Instead of using, say, DSM-5 criteria for schizophrenia, can we look at a functional phenotype and maybe group patients into treatment groups and maybe have more success in treating people if they're if they get the right treatment at the right time? So this is where I'd like to see this go clinically. But we can build these connectome-based predictive models, we call it, to allow predictions of scores or behavior. And I think, actually, even though I only show one matrix here, and all of these papers tended to use different data and different models, I think from an individual connectome, you should be able to get all of this information once you have these models. So for an individual, you could get their connectivity matrix, and from that, potentially get a complete profile if you had models for all these different constructs that you were interested in. And so part of the work right now is trying to build these models and learn how best to do that. So how do we do the most simplest form of this connectome base, the CPM, is to look at what edges vary as a function of behavior across subjects, and then build a model based on that and bring in a new subject and apply the model and see how well we do at predicting their score. So the simplest way to do that is to stack people up like this. We get a matrix from, you have your subjects 1 to n-1, let's say, and you have a connectivity matrix for each subject. And then in this case, we have a behavioral measure, let's say it's D prime, so D prime score from an attention task. And you have a score for each individual. And you're basically going to correlate these scores with the matrix, and you're going to then pull out the edges that either vary positively or negatively in that correlation as a function of D prime. And so you're using this as a feature extraction step to isolate the edges that are relevant for this behavior. And this could be behavior, a trait, a symptom, anything like that. And so you get a mask then, essentially, which is like this. And you could then look at the positive tail of that, the negative tail, and you could sum those edges, and you could get a score for each individual that gives their network strength, if you call the sum of those edges network strength. And because we defined it this way, the network strength increases with higher D prime scores. So then you can fit a line to that data, and that line becomes your model, such that you can apply this mask to a new individual and calculate their network strength. And then you should be able to, from this model, once you have their network strength, you should be able to predict their D prime score. And so if we go through and leave one out and cycle through and do that, here's the first subject. Can you see that dot? No. With the first subject, there's a dot. And that's so we calculated their network score, and we can then look at how well we did in terms of predicting their D prime from the network score using this linear model. And then you can cycle through a whole bunch of subjects and do this and get some sense of performance of the model, which is really how well do we do in predicting the scores versus the observed scores. And you can do that for the positive edges, and you can do that for the negatively correlated edges. And you can combine these in a general linear model, and that general linear model gives a similar result in terms of performance. So it turns out it doesn't seem to make much difference whether you use the positive model, the negative model, or the combination of the models. The performance is pretty similar across these. Now, one of the limitations of this is that that summary statistic there is probably a weak way to build these models because we're losing a lot of information. And if you can keep some of the information separate, you might ultimately do better. And so we're looking at other models that do that. The other thing is that I've been talking about having a resting state scan. We've shown recently that actually resting state is probably the worst condition in which to collect these connectivity matrices in. But if you have task data, you can actually build much better, or models that perform much better in terms of prediction. And so we can do that. And our cross-validation has gone from now leave one out to like split half. So if you have 800, when Emily first did this, there were like, it was the first release of the HCP, there were like 118 subjects or something. Now there's like 1,200 subjects, and 800 of which are good. But you can do, train on 400 and test on the other 400, and it works pretty well. So we had two basic papers on this. The first one by Emily on the functional fingerprinting, let's say, and then Shilin, and this model is available. You can get our atlas, and you can get the code for this from our GitHub there. So that's kind of the basics of that. And as I said, there's lots of behaviors that people are working on, and there's lots of prediction. There's 2 reviews that are coming out, one by Steve Smith's group and the other by Mike Millam and some others whose name I'm blanking on, Gail Veracqua, who looked at 1 million different atlases and modeling strategies and their predictive abilities. And it's those 2 papers, if you can find them. I think one's published, one's on bioarchives, and I've given them to some people here, but Jim knows them. You can look those up at the different techniques for doing this, and they are making progress. But the bottom line is that there's a lot of information in this matrix, and it's cool to be able to extract that information and learn what information is in here. And I think we're just at the tip of the iceberg. When there's 70,000 connections there, that's a lot of data, and we need to learn how to read that data. But I want to go back to the beginning, which is this, the ingredients for this was an atlas for node definitions and an fMRI time course data. I'm going to focus actually for the next little bit on this atlas. And the motivation when we started on this was, could we, is there a better atlas? How many nodes is the correct number of nodes? And when I actually originally worked on this, I wanted there to be a functional atlas that was kind of equivalent to the cytoarchitecture atlas, Brodmann areas, or there's been other anatomic atlases like the AAL atlas out of Montreal. And now there's lots of these functional-based atlases based on fMRI data. And there's a ton of interest in this atlasing work. Like, how do we, what's the ideal way to parcellate the data and divide the brain up into these functional atlases? So you can see there's lots of papers and lots of interest in this. And there's tons of citations. So you can actually do, it's funny, you can do an H-index on topics. So the H-index there, 163 peaked. But when I started on this, I had a postdoc, Shilin Shen, who I said, there's the Brodmann atlas, and then there's going to be the Shen atlas, and the Shen atlas is going to be the functional atlas, the equivalent to Brodmann areas. And this was back in 2010, we did our first one, and actually lots of other people had done atlases by then too. We weren't the first at this, but I was kind of psyched about this, using functional connectivity to define an atlas. And so this is based on the same concept of resting state fMRI And if adjacent voxels are oscillating together, then they're probably part of the same node. That's the basic assumption there. And so if they have high synchrony, we'll cluster them together, and then we'll divide that into various subunits and try to divide up the brain in that way. So we've had a couple of papers on this, and the 2013 atlas is the 268 node atlas, which is the one that's pictured there. That's available from NITRIC, the NIH website. And we actually have a 368 node atlas. Those seem like odd numbers, well, even numbers, but strange numbers. We actually put in that we wanted 300 nodes, but we got 268. And then later we put in we wanted 400 nodes, and we got 368. So that's where those numbers come from. But anyway, so you can get these atlases, and there are lots of other atlases out there. In fact, here's some of them. So Cameron Craddock has an atlas of actually Cameron Craddock has a bunch of atlases. There's one that's about 400 nodes. Thomas Yeo has an atlas, or he's got a few atlases out. The Matt Glasser one is high profile because that was in Nature, I think, and that's a multimodal atlas. I forget how many nodes the Power Atlas is. How many nodes are in the Power Atlas? Does anybody know? 264. And the Gordon Atlas, which is 333. So you can use any of these atlases, and in fact, it turns out that in these predictive models, they're not because of that collapsing the data into a network score, it's not super sensitive to the atlas. But I think that's actually a problem with the data reduction strategy of the modeling. And if you handled the data better, they might be a little more sensitive to the actual atlas. These are not random atlases. These atlases are a little more meaningful than random, although that's hard to evaluate. It's also interesting to note the number of nodes. There isn't a right answer on how many nodes there should be. That's typical. It's a factor of 400 is a factor of 4 almost greater than the Brodmann areas. And people seem to be comfortable with that. And I'll leave it at that. We'll come back to that point a little bit later. But you can think of this as either these are meaningful subunits, and we should talk about what they mean, or you can think of this as a data reduction strategy. If you think of it as a data reduction strategy, it doesn't matter which atlas you use. But I'm going to talk about these as meaningful subunits. So all of these atlases are based on resting state data. Glasser's atlas is resting state plus some other stuff. Actually, they have tasks. They treat everything together and kind of collapse resting state and task data from the HCP project into this atlas. But most of these are not multimodality. Most of them are resting state. And so, at the beginning of this, I'd said to Shilin Shen, we're going to have the Shen Atlas, and it'll be really cool. You'll be famous. And it turns out now, though, that I've completely given up on that, and that I'm going to tell you that there isn't a fixed functional atlas, and that there's no such thing as a functional atlas. The brain is very flexible in its organization, and these node definitions are brain state dependent, okay? So if you see this atlas under different conditions, it's going to look different. And that's what I'm going to show you data on now. And I'll show you data on now. I'll give you my editorial comments later. Consider parcellation under different brain states. If we're going to look at parcellation, one of the things that kept me from doing this for a while is that most of these algorithms don't have a way of keeping track of node correspondence. And so you need, if you're going to compare atlases across brain states, you need to have the same number of nodes for one thing. Otherwise, you end up with a node correspondence problem, and you don't know, if things are rearranging, you don't know which node rearranged to what. Or it's a very difficult problem to solve. But anyway, so we have an exemplar-based parcellation approach, which maintains node correspondence. So if we start, and basically we start with something like a 268 node atlas or anything. And from there, we develop exemplars, and then we grow those exemplars to fill the space, basically. And so we can do that under different conditions, and we have a way then to compare nodes across those different conditions. So we need an algorithm that, and I don't think any of the, as part of the review process for this parcellation paper, they were like, well, why don't you compare why don't you do this with some of these other algorithms as well? But you can't, because those algorithms don't have a way to keep track of which node goes with which node. And so the comparison becomes problematic. So you need that. So we start with a group atlas. We actually started with several group atlases, and I'll show you results where we did this from 200, 400, 1,000, and then 5,000 nodes. And the result kind of holds across all those different scales. But basically, you start with some random group atlas, and then from there, you can look at how that atlas deforms to an individual, and then how that individual atlas deforms with brain state changes. And so we applied this to data collected under different conditions. We had 6 continuous performance tasks and 2 rest runs. So we had 8 different runs, basically. And we could run this atlas on each of those individually. They were each about 6-minute runs. And we had a TR, I think, of about a second. So 360 volumes per condition. And then what we did was we imaged me 30 times over about 9 months. And so we have a single subject now imaged 30 times. And there's no anatomic variation here. So we're not introducing any, and hopefully my brain wasn't changing anatomically a lot. But we're presumably there's not a lot of degeneration over 9 months. But we imaged me 30 times over 9 months. And this data, I think we've loaded it on Open Neuro now. And so this data is available. And there's also in-magnet performance data for the tasks and things like that, which I'm a little embarrassed to say I really suck at the NBAC task. So 30 times over 9 months. So what we could hypothesize then was that the parcellation should be consistent from day to day or session to session within a condition. And we wanted to see, is it going to vary by condition, so by task. And so this should be a pretty clean sort of setup. These were the tasks. So we had an NBAC, a 2-back task. We had the emotional eyes task, which is the same as in the HCP. We had a reward task, which was kind of like a card guessing task. We had a response inhibition, like a go, no-go task. Watched movies, so everybody's been asking me this. Princess Bride, Up and Away, and Inside Out. Little 2-minute clips of each of those. And so those are the movies. This is Monica Rosenberg's attention task, the grad CPT task. So you see city scenes coming on very quickly, and you keep pushing as long as there's a city scene, and then when there's a mountain scene, you withhold your button press. So it's actually a little bit of a response inhibition task as well as an attention task. And it's trivial for the first few seconds, but it drags on at about the 5th minute or something. It gets a little boring. So there's a attention task, and we did rest at the beginning and rest at the end. And to be honest, we haven't really directly compared the time effect of those rest. The rest runs are pretty similar to each other. So all of these are continuous performance tasks. They're not blocks. In the HCP data, the task data there is block design. So there's rest, and then there's blocks of activation, and then rest. These are completely, so if you're doing 2-back, NBAC, you're doing 2-back, NBAC for the whole 6 minutes. So we think of that as a brain state manipulation. It's a well-controlled way to have the person doing something. And if you parcellate all these brains and you arrange them in one of these tensor sort of modes based on similarity, the movies condition is in the upper left there. And it was actually the most distinct in terms of the node definitions were very different in the movie task compared to some of the others. But there's some things that well, and that makes sense. And there's some other things that make sense, which is that like the grad CPT task and the NBAC task are a little bit similar. And the 2 rest tasks are there next to the movie conditions, and they're somewhat similar. And so that's kind of the bottom line. We do, fundamentally, the answer is going to be yes, we get a different parcellation for each condition. And these are super reliable across days within a condition, and they're super different across conditions. And we can look at this a bunch of different ways. One way is, well, there's 2 ways here that are on this slide. This is looking at parcellation similarity. On the left is measured with Hamming distance. It looks like I cut off, I'm sorry, I cut off the label there. That's Hamming distance. So basically how many voxels overlap in parcels. And the other is node size similarity. So remember, we have correspondence between nodes. And so we know we have a node size vector, let's say, that's 268 points long, if there's 268 nodes. And we can measure the size of each of those nodes, and we can correlate those vectors. And you can see, for example, one of the interesting things is that the movie condition is very different from all the other conditions. So on the diagonal, you have within condition kind of similarity, and that's high. And that's what this histogram is. So this is within condition similarity scores for either correlation of the node size vector or for Hamming, 1 minus the Hamming distance. And you can see, for example, that the differences between conditions, the movie condition is very different from these other conditions. And that maybe the stop signal task and the card guessing task are somewhat similar, right there.
Wait, so were you able to estimate the number of functional nodes that exist for each task to see?
No, so we're starting with a fixed number of nodes. So we did this at, we start with a sample atlas, right, at 200, in this case, 268 nodes. And we're not allowing the number of nodes to change. We're only allowing their shape to change.
So you were saying earlier that if two nodes are fairly similar in their activity and they're nearby, then they're clumped together as a single thing? So
well, we
can
grow
use that to estimate the number of functional nodes?
Yeah, we've tried that, and the answer is basically no. That curve is very, very flat. There isn't a, so we've tried to look at this, and other people, I think Thomas Yo has looked at this too. Is there a happy number that seems to be the right number of nodes? And that's a super smooth curve, anywhere from 200 to 5,000. And there isn't a good way to draw that line.
To say, because different tasks actually require a different number of functional nodes.
Yeah, and that may well be, and we didn't allow that here. So all we're allowing here is a change in the shape of the nodes. But yeah, it could well be the case that maybe there should be 300 nodes for task A and 250 nodes for task B. And we didn't allow that option, but that's yet another level of flexibility that we didn't consider. Yeah.
In scan sessions, were all the tasks? I'm assuming you're watching the movies in the same order, you're not scrambling them, but are the tasks?
The movies never changed. It's Princess Bride first, but the order of the tasks did change. So we always started with rest, then the order of the tasks was randomized, and then we always finished with rest.
And then within a task, was the sequence of the trials the same?
Yeah, no, they're randomized too. You couldn't, I didn't learn the pattern for the attention task or anything like that. They were always randomized, yeah. That's a good question. But you can see from these distributions, they're very different within versus between, and that's the main point here. And this is for the 268 node atlas. And we could, as I mentioned, we could convert that 268 node parcellation to a vector representing node size. And it turns out that that node size vector is actually informative in terms of we could predict which condition we were in based on the node size vector. So in other words, the node size vector is like a signature for that condition. And so we could do classification here based on, we could basically tell with 70 to 80% accuracy, except for maybe in the stop signal task wasn't so good. But we could tell with pretty good accuracy which condition we were in just based on the node size vector alone. And so that is suggesting that it's reproducible and it's unique to that condition. We took this a step further, and some of these tasks actually had behavioral data in them. So the NBAC had some behavioral data. The grad CPT had a D prime, stop signal card. All these had behavioral measures in them. And across sessions, then we actually tried to predict performance. And we could do that with some accuracy, suggesting that the more my atlas looked like it should, the better I was doing in that task or something like that. That's basically what that's saying. And so again, that suggests that these node sizes have some useful or meaningful information in them. So then you could ask, well, is it just my brain is weird? The answer is no. There's Midnight Scan Club data available. So there were actually 10 subjects scanned 10 times, but 2 of the subjects didn't have all the data. Or there was quite a bit of motion. So we used 8 subjects scanned 10 times. And that kind of replicated the same finding. They have kind of these, there's 3 groups here. And these are really, they have 2 semantic coherence tasks. They have these memory tasks, and then they have motor tasks. And I think that's what these 3 groups are in the histogram here. And the data is a little noisier. So they were only scanned 10 times. Mine was 30 times. And I think they have less data per run. And some of these runs are pretty short. But there's still the overall effect of, there's more within task similarity than between task similarity. And that these tended to group by task categories, so these semantic tasks group together. So that would suggest it's not just me. And then, but this is still within subjects. So this was, even though there's 8 subjects here, we did the analysis within subjects. So you're looking within a subject at their parcellation similarity across the conditions, but we're not looking across subjects here. We're just summing the data across subjects. But then we went to the HCP data, and you could say, well, now we're going to introduce anatomic variations, and we're going to go across, this is 514 subjects, only scanned once, but the bottom line is, does the atlas look more similar in a condition across subjects than it does in a, between conditions kind of thing. And this also, strongly, the histograms are pretty well defined here with 500 subjects. There's a strong within subject parcellation similarity, and between conditions, it's not as strong. So this would say that even when you introduce anatomic variations, we still get a better functional parcellation that looks more similar within a condition than between conditions. So I would say it's not unique to my brain. And actually, in both the Midnight Scan Club data and in the HCP data, we could predict which condition with some accuracy, which condition you're in based on a node size vector. So we can do prediction in there as well, meaning that these node size vectors are unique to those conditions. And so that's kind of interesting. So then you could say, well, is this variation really, there was a question about, are the nodes, could you combine nodes or split them? And you could say, well, maybe, what's happening here is that the nodes are, conglomerates of multiple nodes, and maybe you're just splitting them in different ways because the resolution's too crude. So we cranked it up. We did 1,000 and we did 5,000. And ultimately, if you get to the level of the voxels, there's no further divisions, right? And so it would plateau at the level of the voxels. But we went up to 5,000 nodes, and even at 5,000 nodes, we see this reproducible rearrangement. And so that's shown here. This is back to my data now, looking at a parcellation with an atlas of 5,000 nodes here. So to do this, we actually evenly randomized like 5,000 seed points over the cortex and subcortical structures, and then we just grew those. And the same starting seed points for all conditions. But clearly, the parcellation is more similar within a condition. And so even at 5,000 nodes, the nodes are rearranging their size. So maybe at 10,000 nodes, this would stabilize and not change, but that's getting pretty high. So what does all this mean? So I think what this means is that state-dependent node definitions should be considered. And in most work to date, people use fixed atlases, and they do look at connectivity matrices under different conditions. So you might consider the fact that those atlases aren't correct for all those different conditions. Now, it might also be the case that it's not critical to much of that analysis, but you should know that they're changing. We are now moving to kind of doing individualized atlases and also state-dependent atlases. So that's individualized on 2 levels. And I'm currently investigating to what extent this influences the results. But the bottom line is that there's no functional, there isn't a fixed functional atlas that's analogous to the Brodmann areas, or to the Brodmann atlas. And so I've kind of given up on, having Shilin Chen define the ultimate functional atlas, although most of my colleagues in the parcellation field haven't, and they're still working on that problem. But this also has, when you start to think about what this means and why this is happening, it does have implications for brain mapping and the problem of brain mapping. And so I'm just going to spend then a little bit more time on brain mapping. So what's the goal of brain mapping? And I've already talked about this with a bunch of the people in here, but the goal of brain mapping is to take a region and to assign a function to that region, right? We want to know, what does this region of the brain do? And that's fundamentally what brain mapping is. Can we assign function to a brain region? But there's an implicit assumption there. Maybe it's explicit, I don't know. There's kind of an implicit assumption about a one-to-one mapping. Also, as an aside, I think we talked about this a little bit, some people. We want to know what each region of the brain does. That comes from task-based fMRI and from lesion studies and things like that. It turns out when we do these CPM analyses and VPA and stuff like that, you see that actually the whole brain is kind of involved in tasks. And we get these complex connections that are not usually observed with task-based fMRI, but they appear to be meaningful, and they do help these predictive models. And so maybe the brain, as an aside here, maybe the brain's a little more complicated than a single region activating under a certain task. But that's kind of an aside. That's a whole nother talk that I have. But what if there's no such thing as a fixed brain region? And this is where I'm going to go. I think there actually isn't a fixed brain region. But the field of brain mapping is, and you could maybe convince me that I'm wrong on this, although I think this is true. The field of brain mapping is looking for a one-to-one correspondence between a region and a function. They want to assign a region a function. And I'm going to make the argument here now that that's not actually true. So it's probably much more complicated than this one-to-one thing. We know from the brain map or Neurosynth databases that if you look at various behavioral domains, and this is our 268 node atlas, and we could go through and look at what behavioral domains are associated with each node in the atlas. And you can see that almost every node has associated with it a ton of different behavioral domains. So one of the arguments is, well, the behavioral domains aren't defined properly, and if you have the right behavioral domain property, you would have the right, there'd be a one-to-one relationship between node and behavioral domain. But I'm going to argue that's not the case either. So this is just blowing up that bottom axis so you can see what those domains are. But let's go and look at this a little bit deeper. So we'll do some math here, some rough math. So let's say there's 16 billion neurons in the cortex. I got that from Wikipedia. The Brodmann atlas has about 100 nodes. The functional atlas, let's say 400 nodes. So if we do the math, 16 billion divided by 100 is like 160 million neurons per Brodmann area. And 16 billion by 400 nodes is about 40 million neurons per node. So when you're using a 400 node atlas, you're lumping together 40 million neurons, let's say, okay? And if we're assuming that we're going to assign a single function to that, we're saying that 40 million neurons are all doing the same thing. And I think most of you would agree that that's probably not the case. At 4,000 nodes, there's still 4 million neurons per node. So that's a lot of neurons in there. And the fundamental question is, do these 40 million neurons all do the same thing? Because to assume that there's a fixed node with a fixed function is to assume that that's the case. In our 268 node atlas, there's about 60 million neurons per node. So I do think that this is kind of the basic assumption of human brain mapping, although maybe we're not brain mapping at the level of a 400 node atlas. Maybe you're looking at smaller regions and things like that. There's still this fundamental assumption that whatever that is, if it's bigger than a voxel actually, I have this on another slide, a 2 millimeter cube voxel has about 85,000 neurons in it. And so even at if you're only mapping a 2 millimeter single voxel, you're still assuming that those 85,000 neurons all do the same thing. And I would say that's not true. But you see this time and again in meta-analyses with BrainMap Neurosynth, in the work on ontology, so coming up with new labels. They're still trying to assign a new label of function to a single region. And we see this in the parcellation work where people are still trying to come up with the ideal parcellation. So if we look at an example of a 6x6 voxel node, so this would be equivalent to a 2,360 node atlas with a maximum of 4 neurons per voxel, let's say. I could have those neurons tuned to different things. Like you could have some of the neurons, these are all, let's say, pyramidal excitatory neurons. They could be tuned to some yellow task component or green or red or black. And depending on the inputs and the outputs, there's going to be this slight different tuning. And if I group things, if I do a parcellation analysis and I group the neurons under these different conditions, I'm going to get different regions defined here. So I'll get the yellow region, the red region, the green, the gray. Maybe the only region that overlaps or is the intersection of those is that little blue region. But probably if I had more task conditions and different inputs and outputs, that would get smaller and smaller. This is incredibly flexible with only 4 neurons per voxel. And I'm not saying that these neurons have completely different roles. I'm saying that they're slightly different tuning. And we see this in the visual cortex. So one hemisphere, V1, has something like 140 million neurons in it. And we know V1 has ocular dominance columns, there's retinotopic maps, there's all sorts of different organization levels within V1. And that's what I'm saying here is in the cortex, some sort of organizational level that may be tuned. You know, maybe you get the red area if there's no outside stimuli and the person's well rested. Maybe you get the yellow area if they have an itch simultaneously while they're doing the task. And if they all of a sudden think about what they're doing in the magnet or have to do outside the magnet while they're doing the task, you get D or something like that. So these are slight differences on the inputs, outputs, brain state changes that influence the subgroup of neurons that's tuned. This is only 4 neurons per voxel, and I can get this sort of complexity. And in general, we have 85,000 neurons per voxel. And I'm sure they're not all doing the same thing. So to me, this seems to make a lot of sense. But what we have to incorporate then in our models when we're trying to assign function to a region is we have to have flexible function and we have to have flexible region definitions. And we have to allow that those regions can overlap and be very, you know, maybe there's an infinite number of combinations of these regions and tuning and function. The brain is complicated. And I think we've been looking at this in a much oversimplified way. For reference, up there in the upper right, that's what an atlas with 1,041 nodes looks like. And you can see those are getting fairly small, although they're still pretty big. And the example I'm giving here of a 6x6 voxel node would be for a 2,360 node atlas. So each of the nodes would be half the size of what you're seeing up there in the upper right. Although you can see those aren't evenly distributed in terms of size. So this is like I started off, I wanted to find the ideal parcellation. I found that the parcellation changes with task. And when I think about it, this is kind of what I come up with as an explanation of why it changes with task. And it seems to me to make sense. There's evidence for this in the visual cortex. There's evidence for this in the motor cortex. There's evidence for this in the literature, in the macaque. There's face regions, and there's regions that are sensitive to different aspects of face processing. And there's all kinds of evidence for suborganization, subspecialization within regions. And there's no reason to think that we can't be sensitive to that with functional connectivity data under these different conditions. And so I think that's potentially why we're getting these changes in the node definitions as a function of brain state. So the neuroanatomists don't like this. And it's really a difference in emphasis. So some of the neuroanatomists would argue that, well, yes, there's functional specialization, but we don't want to break that up and because it's potentially complicated. And yes, I would agree that it's potentially complicated, but I also think that's potentially interesting. And for us to make advances in this brain mapping problem, we got to get away from the assumption of one-to-one mapping between a region and a function. And I think if we allow flexibility in terms of the region definitions and the function definitions, it gets a lot more complicated, but it's probably a lot more realistic than just assuming. If we stick at the level of cytoarchitecture, let's say, and we stick at the level of V1, we know V1 does vision. And the question is, do we want to get any further than knowing that it does vision? And I think to get further, we have to allow for organization at different levels. And so that's my argument there. So the supporting evidence, in MVPA, we know that there's lots of suborganization. We can do hyperalignment based on that suborganization. But even there, there's 85,000 neurons in a voxel. Maybe MVPA or hyperalignment would be different under different brain state conditions. And I think you guys should look at that. I would predict it is. But it would all be useful. It's just you would have a different hyperalignment as a function of whatever you were tweaking in terms of changing the brain state. I already mentioned the visual cortex. It's organized in all sorts of different ways. And we're doing some experiments now. We have, from Brain Initiative, we have some funding to do this mouse imaging experiments. You can do 2-photon single-cell calcium imaging, and you can look at individual cells. So this is following an individual cell here and looking at its fluorescence over time. These are each individual cells. Here's their plots of delta F over F. That's the fluorescent calcium signal over time. And you can look with mesoscopic calcium imaging done at the same time. You can look at the cortex, and you can see these motifs that are in synchrony with the different cell types. And so you can actually relate these kind of macroscopic patterns of activity to single cell imaging. And we also have experiments underway where we can do this kind of mesoscopic calcium imaging simultaneous with fMRI bold in a mouse at 11.7 Tesla. And so we're starting to get some data on that too. But with that, I hope to be able to link individual cell results to kind of macroscopic activity. The other thing about this, and I'm not sure if this example kind of really shows it, but basically these cells are in this tiny chunk of cortex here. These are almost adjacent cells, and they can have very different projections at the cortical surface. So again, showing that they're not all doing the exact same thing, and there is tuning, and that would be supportive of this sort of model that I'm proposing. And also just from the literature, in the macaque, there's regions that respond to face, to color, to disparity, body. And so there's subspecialization in lots of cortical regions in both humans and animals. So this, and then there was a paper, there's this cool paper by Thomas Yeo in Cerebral Cortex 2015, where they were looking at association cortex, and they did kind of a meta-analysis on a bunch of brain map data. I think they took 1,000 studies or something like that, and they were developing this new ontology to look at mapping these new functions onto the brain. And they actually showed these overlapping regions from this new ontology. So they would get isolated functional regions that were associated with different cognitive domains, but these were overlapping regions. And so they went on to say that that's all related to the underlying connectivity, but they didn't take it that next step to say that, well, maybe we need functional, flexible definitions in terms of brain regions and things. And so I think that's important. What I haven't done here, so the point was brought up about, did we consider letting the atlas change the number of nodes? We didn't. We also, in this theory here, I'm not even discussing different cell types, okay? There's tons of different cell types within a brain region. And I would suspect that actually, we can measure with calcium imaging, for example, we can measure excitation profiles of those different cell types. And I wouldn't be surprised if we actually get different atlases for different cell types. And they wouldn't necessarily overlap in the same sort of way. So there's a cell type effect, and MR, so functional MR is independent of cell type. It's really responding to metabolic demand. And so MR is some complex integration of glial activity and excitatory activity and things like that. Whatever consumes oxygen leads to an MR signal. And so there's probably some overlapping cell type information there. And you saw from, or maybe you saw, Emily Finn has a paper out on layers, and you can look at inputs and outputs. There's probably some organization that differs across layers too, and we haven't considered that at all. So I think the bottom line is that it's probably, we're working maybe in a little bit of an oversimplified realm, and that we have to consider some of these factors in looking at the brain flexibility. So I want to say, I'll just wrap up with, I think the brain is pretty complicated. There isn't a single functional parcellation. These state-dependent parcellations reveal flexible organization of nodes, and I think that flexibility is fascinating. And but unfortunately, most neuroimaging studies ignore this complexity and flexibility. But as we make more and more progress in terms of linking brain organization to behavior, I think we may need to take some of these things into consideration. And I'm not saying that the infrastructure is changing in any sort of way. You can have a fixed infrastructure and still have functional flexibility within that. So this doesn't discredit or discount any of the structural work that's gone before us. This is within a fixed infrastructure. The brain can have flexible functional architecture. And so with that, I'd like to wrap up and thank, there's a bunch of people. That's a pretty old picture now of Emily and Monica and Marvin Chun and the gang at Yale. In fact, that's an old trio, I think, 3T trio in the background. No, in fact, that's a 1.5T sonata in the background. But there's a big group of people doing this at multiple levels, and I actually do very little of this myself. This is kind of everybody in the lab contribution. So thank you. I'd be happy to answer any questions.
Now I see that I should have not just tried to sell you our software products, but we should have talked about some research. And wouldn't you agree more, although that's more of a preamble, wouldn't you agree more with Ross Poldrack, who states that we're just using wrong terms to describe what we're actually studying, right? Because all the
yeah, that's the ontology thing, is that we're using the wrong terms. But so he has an ontology paper. So I interrupted you. Why don't you finish what you're saying?
And those wrong terms go actually through. That's why in your talk I was like, I'm siding with him, and then I'm about to burst. But usually it's from burst to siding. Let's say, thank you for using parcellation instead of saying atlas.
Yeah.
Right? Yeah. Because atlas, usually when I open atlas, there is objective information presented in that atlas. In our case, we're dealing with nothing objective because we have no clue what we're studying, right? We are presenting random blobs, right, which we parcellate according to some algorithmic criterion. So even if there are regions, we might not even be able to tell, right? So I would say they're maps, like a kid
Sure.
could draw a map.
Sure.
Right? He
Yeah.
cannot draw an atlas
Yeah.
because he doesn't have objective background, right? So even the term atlas is incorrect term to use to describe what we're doing. Functional connectivity, as Bob Cox says, there is nothing functional and nothing about connectivity in that kind of statement, right? We are talking about correlations of secondary order measure which you acquired from the brain, right? So all these as cacophony, like, can we say that there is no atlas, single atlas, with objectively defined regions? We know that there is one with clear borders, right? Can we assign a function? And then you say, yes, we can. Vision, early vision, right? Yes, we can, right? How to tease it apart? How do we prescribe that, oh, within this region we can do more or more specialized kind of description? That goes about ontology, again, using correct terms to describe some objective matter. It's just my opinion that we just cannot fathom to describe higher order functions of those regions we don't even know, right? So it's not a matter of not having such a thing, but us unable or incapable to actually describe it.
Yes. So that's great. So lots of things there. First of all, Yeah, there are lots of people that say there's, like, none of these atlases are right? They're all wrong. And maybe it's a data reduction strategy, although I would say they're also somewhat better than random. I mean, they do have some, you may argue, are you nodding yeah?
Arbitrary. You use that word again, correct word when I was about to burst. Arbitrary.
Yeah.
Not random, arbitrary.
Yeah. And nobody knows what scale we should be at. I do think that, I mean, I like functional connectivity. I do think that, things that are oscillating together are probably somehow in this meta, whatever, doing something jointly, And there's lots of evidence that suggests that, yeah, that's a meaningful measure. Like Bob Cox may make fun of it, but he's totally in the field too. And,
the truth is somewhere in the middle.
Yeah, the truth is somewhere in the middle. But yeah, there are lots of caveats, and I would accept that. I do. So Gail Veriquois, somebody says all these atlases are wrong, and I agree. It's a data reduction strategy, fine, But I do think too that there is when you get to the ontology thing, so Russ Poldrak's view is that our labels like memory or attention or whatever are wrong, and that if we just had the right labels, that maybe we could get in a data-driven way. If we just had those right labels, then we could assign a reasonable function to a region. But that still makes that assumption of a one-to-one mapping. And I've come around to thinking that that's a wrong assumption. There is not one region of the brain with 40 million neurons is not just doing one thing.
What is that? Vision. Makes it fun.
Well, but if you want to say, if you're happy with vision as a description of function, we're almost done brain mapping, right?
No.
We know Broca's, we know Wernicke's, we know the motor cortex. I mean, this chunk over here does motor. We know that. So are we done? No, I don't think we're done mapping. We need to look, we're interested in looking at more detail, right? And so that's when we break it down into these subregions. And I don't think it's see, the question is, do you buy functional connectivity as a measure? I kind of do, and
There's nothing wrong with correlation of functional signals, right?
Yeah.
But that's a term, right? My functional connectivity might be different from your functional connectivity definition. I would use decouple instead of correlation. I would use some other metric.
Oh, sure, but I don't think it's not necessarily pertinent to how you measure correlation or what that measure is, right? You're making some, what did you say?
Well, the directionality. I mean, like.
Yeah, we don't have any, in bold information, we don't have any directional information.
But you could compute directed information, cross entropy. You have data that's long enough that you have probability distributions over
yeah, but
subregions, right?
In bold,
so you can correlate.
You can get that, but it could be erroneous because in bold you're dependent on plumbing and things like that. So like directional information isn't reliable in bold. I mean, we're looking at really slow fluctuations here. They're a complex mix of all those things I mentioned before, in addition to the local vasculature and plumbing and stuff. And so to compare A to B and see which one went on first, that's really difficult to do. And I wouldn't put too much effort into doing that because of the plumbing confound. But yeah, certainly
It's information that's passing from one region to another region during some sort of a task, right?
Sure.
So wouldn't it be nice to be able to quantify like how many bits of information are actually being sent from one region to another region? How much processing is actually going on? What are the thermodynamic costs of all of that processing?
It would be nice to have that, but we don't have that, right? I mean, yeah, it'd be great to have that. I agree. Sure.
Can I ask a different question?
Sure.
Which is, I think, in a way, there are sort of two competing explanations for the kind of observation you have that the membership changes, right? So the voxels, certain subset of voxels will change its temporal profile of its response as you change the task, and that will change its grouping with other voxels, and then the parcel definition. And one explanation is that there's a fixed architecture, but there are these overlapping neural populations that you showed really nicely, right? And so, one task will activate this subpopulation, another task this other subpopulation. So that all makes a lot of sense. you know, but in a way, you could think of the architecture as still fixed. So if you could measure every single neuron in the brain, and that was your atlas, you know, every single neuron in Todd's brain, then it would be fixed, because you know, a orientation diagonal tuned neuron is always going to be that,
Yeah.
right, or something.
Yeah.
The other alternative is that there's actually a change in the dynamic organization, so that the function of the same fixed population of neurons is actually changing based on its connections and intercorrelations. And do you have a strong feeling about?
No, but yeah, I could agree that that could be another explanation for it too, right? Is that it's the same neurons, but they're getting different inputs or outputs, or different inputs, let's say, from other neurons. And that's kind of the second argument, right?
Yeah,
It's always,
neuron, it's a tuned
yeah,
neuron, and then it becomes something else.
yeah, yeah, which is consistent with your positive versus negative response of, or was it you were telling me about no, who was telling me who had a nice positive negative curve? I don't know. But yeah, you can, you change the function of something depending on the state and or the input, and nothing else has to change. Like nothing, like again here, nothing's changing here. It's just which neurons are firing, right? And in which order. And I guess in my case, I'm saying that something is driving different subpopulations, whereas in your case, you're always driving the same population, but in different ways. And both are equally fine, although, yeah, both are equally fine. But I do think that there's some suborganization that we need to consider, and I don't think we're considering that. Yeah?
I came up with good analogy.
Okay, good.
We could discuss it in the evening. So there we go.
Yeah.
So one thing when we were talking about functional connectivity the whole time, I kept thinking about, okay, over some task time or over resting state, you're just taking the correlation of these fluctuations over time. But you know, what are the signals that would come out? What if those things are changing really fast and genes are changing really slow at different time scales, or if they're fluctuating back and forth? Could it be that the functional connectivity measure itself is obscuring, and the reason why you're getting kind of similarities, or how much of that drives it? But if you look at the calcium imaging, it's really interesting. It's like if you correlated all those, some of them are going to correlate really strongly, and then there's going to be a gradation, right, from like zero correlation to some amount of correlation. But what's interesting, if you look at time, you just think it was like binary, exceed some threshold, are they active? There's time points when all three of those, or you know, three of those four neurons are active. There's times when there's 4 or 2. But those might be discrete states. But if you're taking connectivity over time, you get a completely different measure of how things are connected.
Yeah. So the dynamics problem is really difficult, and I showed this in terms of state, but that's only because that's the easiest way to show it. I'm sure this happens at the dynamic level, but it's really difficult to prove that, right? It's pretty easy to say you're in a brain state A or brain state B. I'm sure we're going between all those states over time, and
so I guess, like, did you see any variation when you look across? So I know you showed that, like, with the reliability of the node size and the parcellation and correspondence, but is there consistent variation across the nodes or edges that allows you to classify the states? Meaning that something about that is giving you some information.
Are you asking, is there a set of nodes that defines a state transition or
Well, the state transition is hard, and I agree. I think especially in fMRI, it's hard to do that. But at least you have like your task states from your data that you scanned over and over again. And I think there's a couple of things. So one is like, based on the comment before, if you go smaller, it looks like it becomes much more reliable if discriminating rest in states, which is interesting. But then, could it be that some edges or combinations of edges allow you to discriminate between different states really well?
Yeah, I mean, there would be, so we haven't looked explicitly at that, but I'm sure there'd be a subset of edges that maximally define that state, or mostly characteristic of that state.
And so if you see some structured variation across different task states between certain edges, does that give you any information about what that region's doing or how it's?
Whenever we've looked at that in other studies, that hasn't been very informative. Like, you get that mess of connections everywhere, and it's not like just one network or one system or something like that.
And the last point to this, which is kind of the same thing, just another way, which is what if you do third order, so you have another edge that's modifying another edge? So it's like dynamics affecting each other. And then I know it's combinatorially explodes even more.
Yeah, well, I don't think we could discriminate that, right? I don't think we could tell what's driving, like we can't tell what's driving what.
But then so you have like two nodes that are fluctuating together, but then when those edges fluctuate, that might affect another edge. So it's like the interaction of two things versus the interaction of two things, the interaction of two other things
Yeah.
coupling.
Yeah, we haven't looked at that.
So is that just statistically calculating the 3-point correlation functions instead of the 2 points? Like,
Yeah, I think you
The-
could
the
do it in
correlate.
different ways. So one could be like you have three nodes interaction.
Yeah.
One way. And the other one could be like it's actually how one state affects the state of another, coupling of another at a different point in time.
Oh, at different points in time.
Well,
So
that's
it's not
not
just
the fourth
over.
order, so you can go even further.
That's,
Yeah, I think the whole, moving window and all the temporal dynamics, I mean, I'm very interested in that, and I've had a really hard time getting like reproducible results in that, and knowing, it's hard to know what state you're really in.
I think we should thank the speaker. There's some snacks out there if you'd like to stick around and talk some more.
Great, thank you.